ask some questions. You should participate. Okay. So first of all of you believe that protein purification is an important technique to know and learn. You should unmute. Just unmute and tell. Do you think it's an important technique to know protein purification and identification?
Yes, sir.
If you feel, then only you are here. Otherwise, you will not be attending this lecture, isn't it? Am I right? You feel it is a good thing to know about protein purification?
Yes, sir.
Okay. So once you know how to do protein purification and you purify protein also, but what comes to your mind? What will you do with purified protein? What all things you can use purified protein for? There should be some use of the purified protein. No? You are not just purifying the protein for fun of it. Okay, so there should be some
So
use
to
of
identify
purified
its
protein.
function, to identify
Yes,
its
Shreya?
function,
Yeah, your
to
voice is
identify,
not that clear. It's like kind of resonating in between.
so to identify its function and to create, suppose we have to create antibodies or antigens, we need to purify protein for that purpose.
Okay, to identify its function and to raise antibody again. This is what you are saying, Shreya? To raise antibodies and to identify the function of the protein.
Yes, sir.
Okay. Anybody else? Any other the other utility of purified proteins? What all uses you can put purified proteins for?
Purified proteins, sir, they are also extremely valuable for biochemical reagents as well as for they can be also used for the growth factor like hormones and some enzymes like DNA polymerase.
They themselves are most of these therapeutic uses. There can be proteins of therapeutic use, and
Yes.
I think I think one message. Yeah, there are some for testing diagnostics purpose. Okay, Shushti has written in the chat box that yes, proteins are also utilized in several diagnostic kits. Okay, so wherever you have to do diagnostics, you can use purified proteins. Okay. Okay, so I believe there are many more uses, but all of you are on the same page that it is important to uh, have this knowledge of purifying proteins. Okay, so today we will discuss protein purification methods. Okay, we, as Dr. Gopalji pointed out, we have been, our lab has been doing protein purification since almost 20-25 years, since my PhD, postdoc, and currently also we are using different systems for protein expression and then purification. So we use bacterial system, we use insect cell baculoviral system, and also mammalian system. Okay, all these systems for protein expression and then eventually purification by different techniques. Okay, so we'll just get started. And as I said, as soon as I share my slides, I'll not be seeing you directly. Okay, so feel free to interrupt me whenever you feel that you are not able to understand or you have a doubt or a question. Okay, so just unmute yourself and ask the question, because I'll not be able to see the chat box again and again. So just feel free, unmute and let me know so that things are clear to everyone. Is that fine? Yeah, you should respond. Otherwise, I'll not know.
Yes, sir.
Is that
Go ahead.
get going? Okay.
Okay, okay.
So let me now share the slides and get started. Okay. So is the slide visible to everyone?
Yes, sir.
Full screen. Yeah, now this is full screen.
Perfect, sir. Perfect. Yeah.
Okay. Okay, let's get started. Okay. So yeah, today this is the central dogma. All of you are aware of the central dogma that DNA upon transcription gives rise to RNA, and RNA upon translation, here especially the mRNA upon translation, gives you the protein. Okay, so this is like there are exceptions to it, but normally this is what how it follows. So your RNA will translate, mRNA will translate and give a protein. Okay, and this is what we are looking at. We have protein and we have to purify it. Okay, so this protein can be expressed in different systems. It can be in a prokaryotic system like E. coli. It can be highly this highly regulated system, or it can be endogenous system. Okay, where endogenous proteins are expressed, and then those proteins we are looking to purify. Different functions because of the different proteins they express. So yeah, roughly 25 to 30,000 proteins we have in a given cell type. Okay, and when you are looking at protein purification, amongst that 25 to 30,000, your protein of interest can be only one of them. Okay, so it is really a complicated system where you have to purify your protein from 20 to 25,000 proteins. So it is like just finding a needle from the haystack, isn't it? So to make easier, so to make your life easier, lot of times this protein purification is done after overexpressing your protein of interest. Have you heard of overexpression of protein of interest? By recombinant DNA technology, you can overexpress your protein of interest. Have you heard of this term, recombinant DNA technology?
Yes, sir.
Yeah.
Yes, sir.
So sometimes you do want to purify protein from endogenous source, but lot of time for commercial use or therapeutic use, we do express the protein in higher amounts using recombinant DNA technology by cloning the gene and using strong promoter, and then we get more protein. So then the protein can still be 1 in 25,000, but the expression level is so high that it is much easier to purify it, okay, compared to the endogenous level. So there, recombinant DNA technology comes to our help. Okay, so normally, why do I ask this question already? But these are some of the common uses of purified protein. First is to study the structure and function of the protein, other physical properties, what are the like whether what is it is alpha helical, beta sheet, or what is the thermal stability of the protein, pH stability of the protein. All those physical properties can be analyzed by purified protein. To determine the sequence, if you are working with an unknown protein whose sequence is not known, you can purify and then sequence it to know the sequence. To raise antibodies, so antibodies serve as very good tools for several diagnostics and also therapeutic and industrial applications of the protein. For example, a lot of growth factors and other proteins like insulin, they are used for a lot of patient applications, as well as in the industry. We can add purified proteins like enzymes. For example, lipases are added in your detergent so that your fat के दांत भी निकल जाते हैं क्योंकि you have lipase enzyme in the detergent. Okay, so these are some of the common uses of purified proteins. There can be many more, but these are some common uses. Is this clear to everyone?
Yes, sir.
Yeah, you should respond, so accordingly we move forward. Okay, so whenever you want to purify any protein, we have to make a strategy. You cannot just go and say that today I am in mood of purifying protein and just I purify the protein. You cannot just say that. Okay, so you have to make a plan. Okay, make a strategy to purify protein. So even before you make a strategy, there are some general considerations. Okay, then after considering the general consideration, you make a strategy, then you devise a technique, and then you have to have a method. Somebody is entering. Let me allow. Okay. So then you have to have a method for protein detection. That how will you know that protein is purified? So you should have a method for detecting the protein, and also you should have a method to assess the purity of the protein. Okay. So based on the application, the purity is very important. In some application, you need 100% pure protein. In some application, 90% pure protein also will be fine. Okay. So based on the purity, your assessment of purity, you can say whether you got the desired purity or not. Fine. So now going back to the first general considerations. What are the general considerations whenever we start a protein purification? First is the end use. Okay, what is the end use will determine the expression system and purification strategy to be employed. Okay, so if end use is for human therapeutics, then there is no chance that there can be any contaminating proteins. You have to have 100% pure protein. Also, other things you have to think whether you need soluble protein or insoluble protein also will be fine, like in form of infusion bodies. Also, you have to ask the question how much you want and how pure you want. Also, whether you want to preserve the post translation modifications in the protein or not. Have you heard of this term, post translation modifications? सुना है ये term post translation modification? सुना है सबने?
Yes, sir.
Can you give some example of post translation modification of proteins?
Sir, capping,
phosphorylation,
phosphorylation and
acetylation,
Yes, very good. Okay.
sumylation,
methylation, protein folding, protein cleavage.
Yeah, disulfide linkages. Okay, so yeah, very good. So a lot of time these post-translational modifications are very important for proper function of the protein. Okay, so if you are mainly enzymes, okay, if you are looking at a functional protein which is an enzyme, you have to lot of time you need to preserve the modification. If modifications are not preserved during the purification, then they might be not active proteins. Okay, so this is an important question to ask. Also, on the same basis, do you want to retain the biological activity? So if you are looking for an active enzyme, then your purification procedure should be such that it should retain the activity. Okay, प्रोटीन प्यूरिफाई हो गया 100% प्योर भी हो गया लेकिन एक्टिव ही नहीं है, so what the use? Okay, so it can be potentially used for antibody generation but not for an enzyme assay where you need an active enzyme. Okay, so these are the questions you have to ask when you are starting to think about the purification protocol. Okay, okay, so whenever you are choosing a strategy, characteristics of proteins whatever are known to you, if they are known to you, that will also help you design a good strategy. What are the things which helps? First is the sequence. Okay, if you have the primary sequence of the protein, somebody is again entering. Check. So if you know the sequence, that will help you to know what is the net charge on the protein, what is the whether it's hydrophilic or hydrophobic. All those things will be available to you if you know the sequence of the protein. Second is oligomeric state. If you have information whether your protein is monomer or dimer or trimer, if that information is there with you, then also it will help you in purification protocol. Also modification. If you are already aware which are the modifications present in your protein of interest, and that will help you to devise strategy to preserve the modifications which might be important for the activity of the protein. Also the information of interaction with cofactors and ions, because lot of time cofactors and ions will be important for the activity of the protein. So that information if you have, that what are the cofactors and ions required, you can include those cofactors and ions in the buffers and during the purification process so that you get an active protein at the end. Also, where is the localization of the protein? Whether your protein is expressed in the cytosol, in the nucleus, or it is a membrane protein, this knowledge also will help you to devise the extraction procedure for the purification of the protein. And also this information that whether your protein is in soluble form or insoluble also will help you to devise the strategy for its purification. Fine? Is it clear? All the points are clear to everyone?
sir,
Yes.
can you explain the oligomeric state once again?
Yeah, oligomeric statements, some of the proteins they can exist as monomer, or sometimes they can two monomer units can come together to form a dimer, or they can also form a higher order structure, they can form a trimer or tetramer. So based on what is the oligomeric state of the protein, in especially when you purify, you know what size of protein you are expecting. The primary sequence might tell that your protein might be of 50 kilodalton, but if it is going to form a tetramer, what will be the net effective size of your protein if it is a tetramer?
200.
Yeah, okay. So that is important, because during purification, if you are looking at the peak of 50 kilodalton and you are not getting your protein, then you will be disappointed, because your peak will come for 400 kilodalton, isn't it? 200 kilodalton, sorry.
Yes,
Okay.
sir.
So that's why if you have the information of oligomeric state, that also helps you to design the strategy for purification.
Okay, sir.
Okay. So if you don't know a lot of things about your protein, only you have the sequence of the protein, then also there are several tools which might help you to give more information about the features of your protein. So this is one such tool. There are other tools also, but this is one of the popular tool. It is available online and it is for free. So you can just Google also. You can Google for prod param, and it will take you to XPC website. And this XPC website, there is here in this window you can paste your protein sequence, submit it, and within few seconds you will get this result. Okay, so what all information can be seen in this result? That it will tell about the molecular weight of the protein, so that will tell about the size of the protein, so it will be important for deciding uh, the resins to be used for size exclusion chromatography. In STS page where to look for your protein. Also, it will give information about the PI. PI is the isoelectric point. You know what is isoelectric point? Isoelectric point
Yes,
क्या
sir.
होता है? Yeah,
Sir, it's a pH where the protein has the net charge is 0.
very good. Okay, the pH at which net charge is 0. So if you know that isoelectric point for your protein, then based on your buffer, pH of your buffer you are planning to use for your purification, in that buffer, based on the pH, your protein can be a cation or an anion. Okay, so based on that, you can use that information for cation or anion exchange chromatography. Similarly, sequence information also gives you the amount of hydrophobic or hydrophilic or acidic or basic residues, and that will also give you information of net charge of the protein. And if it is a hydrophobic protein, then you can utilize hydrophobic interaction chromatography for its purification. Also, it will give information about the extinction coefficient. One of the method to determine protein concentration is to measure the absorbance at 20 nanometer. So, which amino acids are absorbing at 280 nanometer in proteins? Which amino acids absorb light at 280 nanometer?
Tryptophan.
Tryptophan.
Tryptophan. Okay, and to some extent, tryptophan के अलावा tyrosine and phenylalanine also. Okay. So based on the composition of your protein, if they are rich in tryptophan or the aromatic residues, they will have high molar extinction coefficient at 280 nanometer. If they are not rich, then they will have a lower extinction coefficient. Okay. Also, this tool will tell about the instability index, where it will tell about whether it is susceptible to protease. Then you have to add protease inhibitor in the buffer. If it is susceptible to high temperature, then you have to maintain low temperature during the uh, process of purification. Also, the pH stability. Okay, so you have to use the right pH so that the protein remains stable. So all this information you can gather by just having the sequence of the protein, and then all this information will help you to design a good strategy for the purification of the protein. Okay. So also, when you are especially using recombinant DNA technology to overexpress your protein of interest, then you have to clone your gene of interest in a suitable vector. And the suitable vector will be also be dependent on which system you want to use for expression. So if you are using bacterial system for overexpression of your protein, then there are several bacterial vectors. If you are using insect cell mammalian system or yeast system or mammalian system, whatever system you are using, you have to choose the right vector, clone the gene in the right vector, and then Also, you have to decide whether you want to express your protein with a tag, affinity tag, or without any tag. If you add affinity tag, that will allow for affinity purification of your protein. Also, affinity tag can improve the solubility of the protein in some cases. And these are some of the affinity tags which are commonly used. So, GST is glutathione transferase. Only is the his tag. This is the flag tag and the sequence of flag tag. This is HA or hemagglutinin A tag, and this is the sequence of the tag. This is MVP. MVP is the maltol binding protein tag. So these are some of the uh, tags, okay, which are called affinity tags. They can be included along with your protein by cloning your gene along with the sequence for the tag, and it is only possible in recombinant DNA technology where you can introduce the tag and the protein will be now having this tag fused to them, and these tags will help to purify the protein. And also you have option to remove the tag by after the purification. If you want that tag should not be there, you can remove it. So for that purpose, normally a protease cleavage site is added between the tag sequence and the protein your gene sequence. There is a protease site added, and that will help you to uh, remove the tag after purification is done. So whenever you are using protein purification techniques, one of the most common technique which is utilized for protein purification is chromatography. Have you heard of chromatography?
Yes, sir.
Okay.
Yes, sir.
Chromatography is the most commonly used technique for purification of protein. So whenever you are going for purification of protein, normally the first choice is a chromatography technique. If that is not accessible to you, or you it is too expensive, then you have to explore other methods. But normally the common method is chromatography. Okay, so normally this is the workflow whenever you are using chromatography technique. That you have a sample, which can be from a cell extract, it can be from tissue or from blood, or it's a purified peptide. All this can be your sample. And whenever based on the sample's properties and the properties of your protein of interest, you can subject it to different separation techniques, which are different chromatography techniques. Can be affinity, if there is an affinity to specific molecule, ion exchange based on the charge on the protein, size exclusion based on size of the protein, reverse phase chromatography based on the hydrophobicity of the protein. And after subjecting to any of these techniques, the question you ask is the protein pure and is the protein active, especially when you are looking for enzymes. Okay, if the answer is yes, then if it is diluted, you can concentrate and then use it. If the answer is no, then you have to go back, modify your strategy, and purify it further so that you get finally you get an answer in yes. Okay, so in this case, after each step you have to assess the protein purity and activity, so you know that you are moving in the right direction. Okay, so this is just a overview, but this is what you have to look for and ask this question at the end. If you are satisfied, then you are done. If not, you have to go back and replan your purification strategy. So before we purify any protein, we have to get the protein out of our cells where we are expressing it. Okay, so it can be your bacterial cell, yeast cell, mammalian cell, whatever cells you are using for expression of your protein. You have to get your protein out of it. Okay, so these are the common methods by which you can release the content of your protein from the cells. So you can rupture the cell wall in case of plant or bacteria, or you have to rupture plasma membrane in case of eukaryotic cells. Also, you can use different detergent which will get the protein out of different organelles in the cells. Okay, and so that this lysis can be achieved by different means, by physical means, by sonication, or by osmotic shock. Okay, so in physical means, you can use freeze thaw method where you freeze your sample. You can rapidly freeze in liquid nitrogen and then thaw in warm water. So this rapid freeze thaw will create water crystals that will pierce hole in the cell wall and cell membrane, and that through that the content of the cells can come out. Also, you can use a sonicator where high frequency sonic waves will create holes in your cell walls and plasma membranes, and content of the plasma membrane or the cell content can come out. Also, osmotic shock can be used by using the, because the plasma membrane is a semi-permeable membrane. So based on the different, you can keep the cells in hypoosmolar buffer, and the water will enter the cell and it will burst open the cell. So based on the osmotic shock, also you can rupture the cells. Also, sometimes you can use enzyme mediated cell lysis. For example, for bacterial cells, you can use lysozyme, and lysozyme will break the cell wall and contents can come out. It can be used to rupture the cell wall, cell membrane, plasma membrane, to get the proteins out in the solution. Okay. Once you have the proteins out, you also, yeah, before this is just the workflow of whenever you are using recombinant protein for your purification purpose. So in this case, you have to, as I pointed out earlier, you have to design expression plasmid based on the system you want to use, and you clone your gene in that, you transform into the suitable host, select using antibiotic marker. Most common antibiotics use the ampicillin and kanamycin. Then your positive clone you have to grow in the culture medium. For example, if you are using E. coli system, most commonly it is used in LP broth. Okay, and then you have to, mostly inducible systems are used. So you have to induce the protein expression. In case of bacteria, most common inducible system used is based on IPTG. T7 promoters are used, and IPTG induction is used for expression of your protein of interest. Once your expression is done, you have lyse the cells based on the methods I listed in the last slide. Then you have to centrifuge the lysate so that the soluble fraction can be collected as a supernatant, and that soluble fraction then can be subjected to one of the column chromatography methods based on the properties of the protein. You can do that, and then you collect different fractions and can check the different fractions to assess their purity uh, by running a SDS page. Okay, sodium rhodecyl sulfate polyacrylamide gel electrophoresis. सबने सुना है एसडीएस पेज के बारे में? Have you guys heard of SDS page?
Yes, sir.
So these are the steps. So in SDS page, once you run the gel, you use a specific protein stain. Can you name that protein stain used commonly used for staining proteins in SDS page? कोमासी स्टेन सुना है किसी ने? कोमासी brilliant blue. Have you heard of this stain, protein stain, कोमासी?
Yes, sir.
So that is one of the common protein stains. So that will stain the proteins, and then based on the stain proteins, you can assess the purity of your protein. Okay. So these are the steps whenever you are using recombinant protein expression and purification of that. Okay. So sometimes also, before we move on to the chromatography technique, it is a good idea to get your sample concentrated in your protein of interest. Okay. So, after the cell lysate, this step where you are centrifuging and getting a supernatant, still the supernatant might still have hundreds of proteins in that. And in that hundreds of protein, your protein of interest is just one among those hundreds of, or sometimes thousands of proteins. Okay. So if there is a method to get your protein concentrated before you subject to chromatography, that will make the possibility of getting pure protein more compared to using directly uh, complete entire mixture. Okay, so these are the two methods in which you can precipitate the proteins and then check which concentration or which fraction your protein is present in, and thereby sometimes you can get almost 10 to 15 fold uh, you know uh, enrichment of your protein. So one method is called ammonium sulfate precipitation, also salting out by ammonium sulfate. So what happens here that ammonium sulfate is a highly water soluble salt, and normally proteins they are also no most of the proteins are water soluble. Okay, so normally that's why you use aqueous water based buffers for protein extraction and purification. So, but the solubility of different proteins is different. Okay, not all proteins are equally soluble in water. So the proteins which have more hydrophilic uh, amino acids on the surface, they are more soluble in water, and while the proteins which have more hydrophobic residues on surface, they are less soluble in water. Okay, so taking advantage of this particular property, this ammonium sulfate precipitation will like selectively precipitate the proteins at different concentration. So whenever you use ammonium sulfate precipitation, you gradually increase the amount of the salt. So normally we start at 30% saturation, we increase to 40%, 50%. So the proteins which are least soluble, they because ammonium sulfate, what it does that is highly soluble in water. So when you add ammonium sulfate in water, what it does, it removes the water of solvation. Okay, so whatever water was keeping the protein in solution is taken up by ammonium sulfate to get itself dissolved in water. Okay, so now proteins don't have water to remain soluble. So hydrophobic patches of the protein surface they come together and they result in aggregation of the protein. Okay, and thereby the protein precipitates out. So this way, you add different amount of ammonium sulfate, you will see that the clear solution of protein has turned turbid. That means that some proteins have aggregated. You can do a centrifugation step. You can collect the pellet. That is the so this is not a like uh, irreversible process. This is reversible. So in the pellet, if you add fresh vapor, it will again come into solution. But what you can do is you collect pellets of different percentage of ammonium sulfate, 30%, 40%, 50%, and then those pellets you redissolve in your buffer and then run SDSPH and stain it. So now you see that initially your protein was in say 50 ml of original lysate, but after ammonium sulfate precipitation, you collect different precipitates dissolved in just say 1 ml and then on the gel, you see that your most 90% of your protein is now present in say 60% ammonium sulfate concentration pellet. Okay, so what will happen that from 50 ml, your protein is now present in 1 ml plus from say 10,000 proteins. Now the contaminating proteins which are present in that fraction is only say 200. Okay, so that way you get 15 to 20 fold enrichment by using this particular step before you subject the sample for chromatography. So ammonium sulfate precipitation is a very powerful tool to enrich your protein before you proceed for any of the chromatography technique. Also except for affinity, because if you are having an affinity tag, that affinity tag itself is so powerful technique that you don't need this enrichment step, because affinity is very specific. So if you have a particular affinity tag, in that case you can directly use the supernatant for affinity purification. But if you are not using affinity purification, then this enrichment step is very important. Also another way to enrich your sample is doing organic solvent, that is acetone precipitation. So this also acts in the same way, but here it is by decreasing the dielectric constant of water and then sequestration of water of solution and resulting in protein aggregation. So in this case, the protein aggregations sometimes they might not solubilize again in water, but again if your protein gets solubilized, then it is good. Otherwise, ammonium sulfate is the way to move forward. So also sometimes dialysis can help to make your sample enrichment or also purifying the protein. So how it enriches your protein? So by removing small contaminating proteins, you can enrich your protein of interest. So for example, your protein of interest is a large protein, say 300 kilodalton protein. So you see that most of the cell proteins will be much smaller than 300 kilodalton. So you can use a dialysis bag with a cutoff of say 250 kilodalton. So what will happen? All the proteins which are 250 kilodalton and smaller, they will move out of the dialysis bag. Only the larger proteins will be retained in the bag. So what will happen that by doing this single dialysis step, you are able to enrich your protein of interest many-fold, because all smaller proteins are out of the bag. So now you can collect the content of the bag that will have your protein of interest and very few contaminating proteins. Also, you can use dialysis to remove salt from your excess salt from your buffer, or you can also use to exchange the salt of your buffer. So both functions can be done by dialysis. All of you have heard of dialysis and dialysis membrane? Yes or no?
Yes, sir.
Okay. Now coming to chromatography. Okay, so briefly we'll see the history of chromatography. So it is the process of technique of separating molecules or components in a mixture, which can be a gas or liquid, according to differential absorption and dilution. Okay, so based on this property, different chromatography techniques have been generated, and we use based on our property of our protein of interest, one of them. Okay, or a combination of them also, not just one. It can be combination of one or more. The first, this term was invented, and the technique was invented in 1906 by Russian botanist Mikhail Tvet. Okay, and basically, chromatography is physical separation of mixture into individual components. So mixture need not be always proteins. You can have different mixtures. For example, mixtures of different dyes can also be separated by chromatography. Mixture of different pigments, plant pigments can also be separated by chromatography. Okay, so it can be any mixture. It can be mixture of gases also. So in that case, we can use gas chromatography to separate. Okay. Uh, Also, chromatography is used in qualitative and quantitative analysis of biological as well as chemical substances. Okay. So not just limited to proteins, other biological molecules like sugars also can be separated and analyzed both qualitatively and quantitatively by chromatography techniques. Okay. This is a basic setup. You can either use chromatography manually by making a manual column like this, or also you can use a machine. I think somebody is trying to join that. Yeah. Right. Yeah. This is a manual way, or you can use a machine. This is an ACTA FPLC machine, so you can use machine also. So manually, whenever you are using, it is cheaper, but you have to put much more effort, because you have to do everything, collect fractions, load samples, keep adding buffer, all this manually you have to do. So here this is the column in which you put your resin. This is the stationary phase in the column. Your buffer will be the mobile phase, and put your sample here. Sample will get separated. You collect the fractions, and you check the fractions by SBS page. You see which fractions contain your protein of interest. While same thing can be done by machine also, but here what is the ease of using it, that it is com- completely programmable. You can use a computer software. You can program the flow rate. You can program the sample volume, and all those things can be programmed. So these are the buffer containers. This is the column in which the sample is. You can there is here, it is not visible, but there is the sample injection port here, where you can inject the sample. Also the buffer, because the pump is there, so buffer flow rate can be controlled by the pump, and they will pump the buffer into the column. Also, the collection of the sample can be done automatically. Here it is not, but there is a fraction collector, automatic fraction collector also can be added to the system, so that will collect the fractions for you. Also, there is a spectrophotometer added in this machine. When the protein samples are eluting out of the column, they will go through the spectrophotometer, and in that, absorption at 280 nanometer is measured. So that also will tell you which fractions have high absorption, and that those fractions have good amount of protein in them. Okay, so this way, the Machine helps a lot, but again, this is an expensive proposition. So if you have money, you can use machine. If you don't have a lot of money, you can use the manual system. So basically, this is a flow chart of all chromatography techniques in general. Whenever you apply a mixture of protein to the column, this is a sample, and how it interacts with the stationary phase of the column will allow for separation of the components. So you see, originally it is black color, and then the components are getting visible again. Hello.
Yes, sir.
Yes,
So
sir.
based on how it interacts with the column, you will have separation of the components, and then you collect the components as different fractions, and then you check the fractions by SGS page to see which fraction contain your protein of interest. So this is a general workflow of any of the chromatography technique. And based on, as I said earlier, based on the properties of your protein, you have different choices of chromatography. If your protein have affinity to some specific molecule, or if you have added affinity tag to your protein, then you can go for affinity chromatography. Okay, if your protein have a high positive or negative charge, you can utilize ion exchange chromatography. If your protein is either too big or too small, you can separate based on the size. Also, if your protein is a membrane protein which has high hydrophobicity, then based on that you can use reverse phase chromatography. Okay. So based on the properties, you can select one of the techniques. Okay. So I think we'll just take 5 minutes break here, and then we get started. Is that fine with everybody? We'll just take a 5 minutes break. Right? So we'll join back in 5 minutes.